area as a hurricane. Um, and the Gulf Coast is kind of reaching up to South and North Carolina, so we're getting some rain right now. But hopefully that hasn't brought your mood down. Maybe if you're like me, you actually like the rain, especially when you don't have to be outside in it. So hopefully you're having a good day, and we'll go ahead and knock out this portion of class, and you'll only have one more block left before Friday. So I appreciate you all being here and being here on time. I will share my screen, and you all can get started on the warm-up. Today is September 17th, Unit 3, Day 4. So when you get into Canvas, you'll just scroll down and you'll find Unit 3, Day 4, and it'll be there for you. Okay, so hopefully that was ample time to knock out the warm up. As I said, I appreciate you all being here. We've got 18 students on the call. That's a good solid number. And those of you who are here, I'm sure are ready to learn. I'm looking forward to you all being engaged in this lesson. I'm looking forward to you all taking yourselves off of mute and sharing or typing into the chat when necessary. But we will go ahead and jump into things. So today we're talking about meiosis and genetic variation. First, before doing that, I do want to review the quizzes that we took yesterday. Keep in mind, folks, that we do want to stay at about an 80%. We want to stay at about an 80% accuracy. Okay, so that means that we should be, on average, as a class, getting A's and B's on our formal assessments. There were some questions, there were actually quite a few questions in this quizzes that proved to be a bit difficult for us. So we're going to go over them now. And I want you guys to, again, be engaged and come off of mute and tell me what you think the answer is. Don't pay attention to how many people answered the question. Just pay attention to what you think the answer is. Okay, so I'll share this tab. All right. So this first question says, cells spend the majority of their lives in which phase? Does anyone know?
Interphase.
Interphase, excellent. And as you can see, our accuracy as a class on that question was only 50%. So that was a bit of a struggle for folks. We want to be up nearer to 80%. And interphase is actually the longest phase of the cell cycle. So this is where cells do spend the majority of their lives. If you are like, I don't know what he's talking about, never heard of interphase, didn't know that it was the longest phase, you should definitely go back and check out our class recording from both Monday and Tuesday when we talked about the cell cycle, and then we also talked about mitosis on Tuesday. I also want to make sure that you all are taking good notes and that you have access to your notes throughout the class period. So we don't write notes for, hand exercise. We take notes so that we can exercise our brains going forward and we can refer back to those notes. So please make sure that you have your notes out and ready to go so you can refer to them starting now. All right, the second question, and our accuracy again was a bit low, 50%. In what phase of mitosis does DNA condense into visible chromosomes?
Is it Anna?
It is not anaphase, but thank you for speaking up, though. Keep doing it, keep sharing.
Prophase.
It is prophase. Excellent. Thank you. Prophase is the first phase of mitosis. And we need to have the DNA condense into chromosomes first before we can pull the chromosomes apart. If they're not in chromosomes, then there's nothing, we can't pull them apart during anaphase. If they're not in chromosomes, then they can't line up in the middle during metaphase. All right, so we gotta have, during the first phase, during prophase, we need that DNA to condense into visible chromosomes. This one, accuracy was a little better, still not at 80%, but it was a little better. Cells of organisms must replicate for blank.
Repair of damaged cells.
That's one of the reasons, yes. So
all of the above.
All of the above was the correct answer here. So cells have to be replicated for all of these reasons in order for organisms to grow, in order for, like you said, the repair of damaged cells to take place, and then also in order to replace dying cells. So all of the above was the correct answer here. Okay. I am the stage of mitosis where the sister chromatids are pulled apart. We did pretty well on this one. Does anyone know? Yes, anaphase. I heard someone say it. Anaphase is correct. Ana, as a prefix, means to pull apart. A, that prefix there, that's a part of the word anaphase, that just means to pull apart. So when you hear anaphase, you got to be thinking apart. That's when the chromosomes literally get yanked from the middle of the cell towards the ends of the cell. All right, this one we did okay on, 57%. What are the phases of mitosis in order? What are the phases of mitosis in order? Someone can just go ahead and rattle off all four.
Is it the second one, the second choice?
Yes, it is. Prophase, metaphase, anaphase, and telophase. Thank you. So we've got to keep that in mind. PMAT. And like I said yesterday, I'll go back while I talk about this. Or like I said on Tuesday, as you all think about some of the skills that can make you a better student, one thing that you might consider is making acronyms for these phrases. So you turn it into something that is easier to remember, a sentence or a phrase. And then you can say, okay, well, this phrase stands for this letter or stands for this specific phase of the cell cycle. I told you in my first year teaching, we had the acronym I play music at the club. I is for interphase. Play is the P for prophase. Music is the M for metaphase. At is the A for anaphase. The is the T for telophase. And club is the C for cytokinesis. So that was a good acronym that my students came up with, but you can obviously come up with your own, my students in my first year. Okay, so keep that in mind. Chromosomes are made up of which macromolecule?
DNA.
Excellent. DNA. That was a pretty good one. We got 71%. DNA is replicated during blank of the cell cycle.
Synthesis.
Excellent. Synthesis. So synthesis takes place during interphase. And we've got to know that that's when the DNA is replicated. So synthesis is literally synthesizing or making new DNA. It's making a copy of the DNA. All right, that's why we call it synthesis. At what stage during mitosis does the nuclear envelope break up?
Prophase.
Excellent. Yeah. Now this was probably our worst question, but prophase is the correct answer here. Okay. The reason for that, again, before anything else can happen, we need to have access to the DNA that's inside of the nucleus. So if we think of the nucleus kind of like a lockbox or a safe, I can't do anything inside of it until I break that safe or break the lock. So I need to have the nuclear envelope, which is the actual box that surrounds it. I need to have that dissolve in order for me to have access to what's inside of it. And that has to happen before anything else can. Okay, so that's the first phase of mitosis. That's prophase. A chromosome that has been duplicated means that, this was a pretty good one, pretty easy one. Someone go ahead and just knock this out for us, please.
There will only be one original.
Ah, what does the word duplicate though? What does that mean?
Oh, copy.
Yes, excellent. Yeah. So we've got to make duplicate means to copy something. So a chromosome that has been duplicated means that there is a copy and an original. Okay, this is a picture of which phase of mitosis? Exactly 50% right.
Metaphase.
Yes, and how could you tell that? How can you tell that this is metaphase? Anybody can answer. What's the clue that this is metaphase?
I cannot tell, but I'd guess it's the blue and red X's in the middle of it.
That's exactly right, Brandon. Thank you. So we've got these X's, which are the chromosomes. We can see that they're lined up in the middle of the cell. So that was the key word that Brandon said. They're in the middle of the cell. And the prefix meta, M-E-T-A, means middle. So in the middle of the cell, we should be thinking metaphase. All right, so that's the clue there. Good job. What is cytokinesis? Didn't do so well on this one. Any thoughts?
The third one.
Yes. So cytokinesis is the stage of the cell cycle when the rest of the cell divides after mitosis is complete. Okay, so I think really I don't necessarily like how that is worded, except for the part that says after mitosis is complete. So mitosis ends with telophase. And then, cytokinesis comes right after that. Okay, so after telophase comes cytokinesis. So that was kind of the clue that that's the right answer in that one. But of course, we do know that cytokinesis means cell movement. Cyto means cell, kinesis means movement. Put it together, that's cell movement. So that means that there are two cells that are moving away from one another. That's what cytokinesis means. That was a tough one. All right, this one was also tough for us. What does not happen during interphase? What is not happening during interphase? Remember, interphase is G1, synthesis, and G2.
Cell division.
Good. Cell division does not take place during interphase. Cell division is M phase. That's what mitosis and meiosis are. Those are both processes of cell division. So that's not happening during interphase because remember, it's interphase, then M phase, then cytokinesis. Good job. I think that was Michelle. Thank you. This was the longest question. You all took your time with this one. Almost done. All right, which of the following shows the correct order of the cell cycle? I'm not even going to spend time on this one. We did well. We've got G1, synthesis, G2. Those are the steps of interphase. Then we've got M phase, which is either mitosis or meiosis. And then we've got cytokinesis. DNA is an example of which type of macromolecule?
Nucleic acid.
Excellent. So the NA in DNA stands for nucleic acid. So that question's got to come to us quick now. You should know that at this point. We did okay on it, not super well, but we got to remember the NA in DNA and the NA in RNA, which we'll talk about more in a couple of units, stands for nucleic acid. And our last question: which of the following types of nuclear division is used for asexual reproduction? Asexual.
The third one.
No, so okay, I think you're talking about fertilization. So fertilization is kind of the result of sexual reproduction when the sperm meet the egg. So we're not thinking about that. What else?
The first one.
Yes, the first one. Good. Mitosis is used for asexual reproduction. All right, so we're going to make a Venn diagram tomorrow. And I really want you all during this lesson and, as we approach tomorrow's lesson to be thinking about the differences between mitosis and meiosis. We've got to have those identified. It's good to know. All right, I'll go back to the slides. All right, so today we're thinking about genetic variation. And genetic variation is an important topic, even as we think about twins, who are people who are extremely genetically similar, but there are even still some differences here. So what do you all know or think about fraternal versus identical twins? Any thoughts on that? Any prior knowledge?
Is it mitosis?
Okay.
It's
We can.
Meiosis, right?
We should be thinking about meiosis or meiosis in this case because we're thinking about sexual reproduction. But I appreciate you, Brandon, for speaking out, and I appreciate you, Shipes, for saying something. Any other thoughts? What do we know about twins? Let me ask you guys this question. Can a brother and a sister be identical twins? Can you have
Yeah.
What'd you say?
No.
No, Michelle, you're right. A male and a female cannot be identical twins because their anatomy is not identical. So as you all know, at this point, I have a twin sister, and that's a question that we get often when we're together, especially when we were younger. Are you all identical? And it's like, definitely not. Thank you for sending your name, DeMarco. All right, so what do you all think about that image on the left? How are those two people twins? They look so different.
Wait, what you said?
How are those two people in that picture on the left? It's on the left side, right? Oh, I haven't stopped. Okay, I'm sorry. I didn't stop. Now you should be able to see it. Those two people on the left, how is it possible that they have the same mom and dad?
The genes.
Excellent. Yeah. So this is all a matter of genetics. And we are about to see a video that really, as soon as I finish with my attendance, that really talks about exactly how that's possible. So I want you all to think about what Brandon just said, because ultimately it is a matter of genetics.
Wait, what was the question again?
What was the what?
What was the question again?
Oh, nothing. I'm sorry. I'm just finishing my attendance.
Mm.
Mr. Rudd.
Yes.
The one on the left is fraternal, right?
Yes, they are fraternal. Yeah. Even though you can actually see there are some similarities between how they look. So let's watch these two videos that explore these concepts.
These pretty teens are best friends, and they're also something else, something that may surprise you.
We're twins.
Believe it or not, they're twins.
Yep, we're twins. We're same
twin
mum,
sisters.
same dad.
You heard right, they have the same mom and dad. What are the odds that one would be black and one white?
A lot of people are just in shock.
Lucy and Maria Aylmer live in Gloucester, England. Maria has a caramel complexion, brown eyes, and thick curly black hair. Lucy has pale white skin, ginger red hair, blue eyes, and freckles. Most people have the same reaction when they hear they are twins.
They say, how did it happen? We don't believe you.
So, how did it happen? Their mom, Donna, is half black and half white. The twins' dad, Vince, is white. The couple was thrilled when a sonogram revealed they were expecting twins.
I just cried. It was shock.
Mom did a double take when she delivered one black and one white baby.
I'm so glad Mum dressed us in such cute little outfits.
Mom dressed her adorable twin daughters in matching outfits, just like twins everywhere. But by age 10, Lucy says she didn't feel like a twin, so why dress like one?
We don't look alike, so why should we have to wear the exact same thing?
Maria says she once wished for Lucy's straight hair.
I used to cry about it. I hate my curly hair.
And Lucy says she was sometimes taunted at school.
They thought I was adopted. You calling me a ghost.
Now 18, the twins not only look different, they have very different personalities.
We're completely different.
Maria loves getting dressed up in chic clothing. She is very outgoing.
I love meeting people. Like, I'm not scared to approach people or anything, so.
Whereas I'm terrified of going up to random strangers.
Lucy prefers casual clothes, but they've come to embrace their uniqueness as black and white twins.
If I have kids one day, they might come out looking like Lucy.
Okay, we'll watch one more.
Now with the twins that we've been telling you about turned social media stars because of the photos showing their different complexions. Their story is inspiring a lot of people. Now their parents are speaking out, and Paula Ferris is here to tell us all about it.
We all love this story, don't we? This morning, the doctors we spoke with said it's a matter of chance. It's so incredibly rare. The mother didn't know if she believed it at first. One looks like mom, the other looks like dad, but both very special. 9-month-old Kalani Dean with light skin and blue eyes, and just 5 minutes younger, her fraternal twin, Jeremy, with brown skin and brown eyes.
I always have them dressed the same because if I don't, people don't believe they are twins.
Kalani looks more like mom, and Jeremy more like dad.
Never in a million years would I imagine having a lighter complected daughter with blue eyes.
One mom, one dad, and twin girls with different skin colors who shared the same womb. All live in Illinois with big half-brother Taylin.
This is a pretty rare occurrence to have two twins that look very different, but it certainly can happen with biracial couples. And it's really just a matter of chance.
The babies are now social media stars. Their story shared thousands of times online.
It's amazing feeling like it's one of those feelings you can't even describe.
But how are twins born with such different complexions? Skin color is determined by multiple genes. Fraternal twins, like the Dean girls, come from two separate eggs, fertilized by two different sperm, and the outcome depends on the genes that are expressed.
By virtue of chance, one twin can inherit more DNA from a specific parent in contrast to its co-twin, and that DNA can code or translate for things like skin color and eye color, and can then result in very different physical attributes.
And Kalani and Jeremy may not always look so different as they grow up. Skin tone can change over time, but for now, mom and dad say their two babies also have distinct personalities.
Kalani, she has more of a personality like her mother. Jay's more just set back, and she's kind of like me, laid back.
Yeah, they are so different in their personalities.
I love this story. You can't help But recognize the deeper meaning here. The parents say what they really want their daughters to know is that you may look different, but really you are the same. You are equals. You're special. You're both cut from the same cloth. You are sisters, and they're equally squeezable, both adorable. But really a great reminder for all of us about equality this time.
It really is. And
Absolutely.
And they had a proud Big Brother. You see Big Brother
Yes,
there in the middle
they
of this cheesing?
did. Really great family.
Thank you.
Okay. So we're there. I think there are two important things to point out, especially from that second video. Number one, this is a matter of genetics. So the genes that you are getting, or the genes that are expressed, are a result of the DNA that you get from your father and the DNA that you get from your mother. So in this case, and as we'll talk about later in this lesson, what we're seeing is a result of genetic variation. It means that some of the genes that are expressed in these two twins, although they have the exact same parents, are expressed a little bit differently. And that there are several reasons, or several possible reasons for that, and we'll talk about them. But then it's also important to note that, as it said in the second video, fraternal twins are the result of two separate eggs being fertilized by two different sperm. So what that means is that you've got two eggs which themselves are different being fertilized by two sperm which are also different. So of course there's a pretty good amount of genetic variation that can take place there. All right. So let's see. We need to select 3 statements that best describe meiosis or meiosis. This first statement says it results in 4 different cells. Does that describe meiosis? It results in 4 different cells. Is that true of meiosis? Hello, am I on mute? No.
I say yes, that is true.
Yeah, that does describe meiosis. Good. So meiosis gives us 4 daughter cells that are totally unique. And of course, meiosis is only producing sperm and egg cells. And so every time that the parent cell undergoes 2 rounds of meiosis, what's happening is that you end up with 4 unique cells at the end. And they're each just a little bit different than one another. That's why we use the word unique. They're different. Okay, this next statement says it undergoes 1 round of PMAT. Does that describe meiosis?
Yeah.
Okay, let's go back and look at this image here. In this case, we can see mitosis has only 1 round of prophase, metaphase, anaphase, and telophase, but meiosis has 2 rounds. Okay, so this statement does not describe meiosis. Okay, this next statement says the cells are identical to the parent cell. Does that describe meiosis? The cells are identical to the parent cell. Is that true of meiosis?
Say that again.
This third statement here, it says the cells are identical to the parent cell. Does that describe meiosis?
Yes.
Okay, let's go back again. All right, so again, we're focused on meiosis. So in this cell, we can see we can see 4 chromosomes. There's a green one, there's a pink one, a purple, and a blue. After the first round of meiosis, the green and the pink are over here, and the purple and the blue are over here. And after the second round of meiosis, the green and we've got even further separation. There's a green and a pink here. There's a green and a pink here. But even these two are different than one another. And then these two are also different from one another. So this statement, which says that the cells are identical to the parent cell, is not true of meiosis. It would be true if this were mitosis, but not true of meiosis or meiosis, however you would like to say it. All right, this next one. Each cell produced is diploid. Is that true of meiosis? Does that describe meiosis? If it's not a diploid, then it has to be a what? Hopefully you all have your notes from yesterday out.
Is that one true?
That one is not true. Okay, so we still need to do some review on meiosis, clearly. But what was the statement? Okay. So meiosis undergoes 2 rounds of cell division to make 4 unique haploid daughter cells. This is a very, very important sentence. If you have not been taking notes, please write this down. And if you did write that down yesterday, maybe you need to go back and highlight it or put a star by it or underline it, circle it a few times. That's very important there. And again, as we approach tomorrow's lesson, we really need to think about the differences between meiosis and mitosis. So meiosis produces 4 haploid cells, and a haploid cell has half the amount of chromosomes. A diploid cell has 2 times the amount of chromosomes. All right, so we've got to be aware of the differences there. So that statement is also not true. Oops, I skipped ahead a few times. All right, so meiosis only happens in body cells. That's not true. Meiosis only happens in sex cells, also known as gametes. Meiosis does produce unique cells, so that means that we know that this last one has to be true as well because we have 3 statements. And meiosis undergoes 2 rounds of PMAT. So I'm going to leave this up so you guys can process this and potentially write it down. I would write down the 3 statements that are true of meiosis. Okay. We'll leave it up for just maybe 20 more seconds. Okay. So genetic variation is really what allows for life as we. Aaron, you have a question?
No.
All right. So genetic variation really explains life as we know it on Earth, how we ended up with all these different species, how we ended up with all these different kingdoms of animals and plants and bacteria. The prevailing theory of evolution states that we've all come from a single common ancestor. So out of that single common ancestor, we've ended up with all these different forms of life. And in reality, we're not that genetically different. We share a great deal of our DNA with fruit flies, which of course we know are basically nothing like human beings, but we share a shocking amount of DNA with them. We are 97% similar genetically to apes and chimpanzees. So small variations in our genes account for large differences in what we become. Okay. So crossing over is the first source that we'll discuss of genetic variation. So you definitely should be writing at this point. Crossing over is the exchange of genes between homologous chromosomes. And this always happens during meiosis. It happens during prophase 1 of meiosis. And you can really see what's going on in that image in the bottom left. We've got a pink chromosome and a purple chromosome. They cross over at the bottom so that they exchange that C portion of the chromosome. Now the pink chromosome ends up with a purple C, and the purple chromosome ends up with a pink C. All right. The second type of genetic variation also always happens during meiosis, and it's random assortment, also known as independent assortment. And what this means is that you cannot predict which DNA has gone into which gamete. So this also contributes to every single sperm cell and every single egg cell. Keep in mind that those are our gametes. Every single one of them is different than the other. So even within the same family, you end up with siblings who maybe look nothing like each other because they got different portions of their parents' DNA. And again, this happens every time during meiosis. Okay. Then the one that you all have probably heard about, our third source of genetic variation is mutation. A mutation, most simply, is an alteration in DNA. There's a change in the DNA, an unintended change in the DNA. All right. Now, we'll discuss mutations in much greater detail in the 5th unit of the course, but some mutations have absolutely no effect on the organism or on the cell. Some mutations have positive effects, meaning they can actually, the organism can survive with that change, or in some cases, the change benefits the organism. And some mutations have deleterious effects or damaging effects that hurt the organism in some way, or in some cases make life no longer possible. And there are many different types of mutations. We'll talk about them in more detail in the 5th unit. I'm not going to go into it right now. But once that mutation is there, oftentimes the mutation is passed down to its offspring. So that's what's important about the genetic variation. It's got to be passed on to the next generation. All right. The 4th source of genetic variation is non-disjunction. And non-disjunction is often the cause of problems. So I'm going to show you this. Oops, I thought I still had it up. Yeah, I do. All right. This image here really shows what happens with non-disjunction. You start off with parent cells that are totally normal and they all look the same. The non-disjunction can happen either during meiosis 1 or meiosis 2. If it happens during meiosis 1, then you end up with 2 normal daughter cells and 2 abnormal daughter cells. If it happens during meiosis 2, then you end up with all 4 daughter cells being abnormal. And what we mean by abnormal is that you can tell a normal daughter cell should have 2 chromatids in each cell. We've got 2 in this cell, 2 in this cell, 2 in this cell, and 2 in this cell. An abnormal daughter cell has either fewer or more than the normal. So in this case, I only have 1 chromatid there, and I have 3 there. And over here, they're all messed up. So this can lead to some pretty serious health conditions. The most common of which, or the most well-known of which is called, not the most common, but the most well-known of which is called Down syndrome. Down syndrome is a result of non-disjunction, in which you end up with an extra chromosome. Okay. And then the last source of genetic variation is fertilization, which is the process by which a sperm and egg cell unite to create a zygote. So a zygote is a sperm and an egg cell together. So fertilization creates a unique combination of genes. All right. Okay, so for the asynchronous portion of class today, I'm going to have you all draw out the cell cycle. So you will need something to write on and something to write with. And I definitely think you should keep this in your notes, and I think it would be a good idea as well if you had different colors, just to make it a little bit, easier for you to see. But I'm going to show you the phases, then you'll draw them. We've got 22 minutes to get through 6 phases, because we've got interphase, then we've got the 4 steps of M phase, and then we've got cytokinesis. So that means about 3 minutes on each picture. All right, so let's go ahead and get started. Interphase. Interphase is very simple, and you guys don't need to make these drawings too complex. All you need to do is a large circle representing the cell, a smaller circle in the middle of the large circle representing the nucleus, and then some squiggly lines in the middle to represent the chromatin. So 3 minutes should be plenty of time for that one. All right, it's been 3 minutes. Prophase is the second phase, or first step of M phase. So here you want to make sure you draw the cell. You want to make sure that you draw the nuclear membrane using dotted lines or lines with spaces in them. And then instead of having chromatin, now we've got chromosomes inside of the nucleus. So draw your X's. Your centrioles, you can draw little cylinders to represent, or little bars to represent centrioles. And then the spindle fibers, you can just draw some curved lines coming out of them. Got 2.5 minutes left to do that. Make sure I got everybody. Two minutes left. About 30 seconds left on this picture. Make sure you're actually labeling it too, please. So don't just draw the picture. You have to include the labels. Okay. Metaphase, which is the second step of mitosis. Just like Brandon told us earlier, we've got the chromosomes lined up in the middle of the cell. So you should draw your X's in the middle. And you can also see that the spindle fibers have now grown out of the centrioles. And so you should draw some of the spindle fibers attached to the very middle of the chromosomes. You can see that all of them are attached to the middle portions of the chromosomes. All right. anaphase, which is the third step of M phase. So at this point, you can see the chromosomes have been pulled apart. They've essentially been ripped apart at their middle. So 3 minutes, please. Again, don't forget to include the labels. Two minutes, 2 minutes. Okay. Two more, telophase. So make sure you highlight and identify and label the cleavage here. The cleavage is where you can see that the two cells, the one cell is starting to become two cells. But also you should draw another dotted line to represent that there is a new nuclear membrane appearing. Okay, and then lastly, cytokinesis. So now you have two totally new cells, and they've got their own nuclear membrane now. So this is the last thing you should draw. Once you are done with your images, once you've drawn all of them, you should go in and write a description of what's happening in each step. Okay, folks, it is 1:20 now. So I am going to stop recording. I'm going to stop sharing